Last lecture, I had started to talk about MHC molecules, and I had stated that T helper cells recognize MHC1 molecules, and that is inaccurate. It's MHC2 molecules that they recognize, and it's T cytotoxic cells that recognize MHC1 molecules. So make that note now. Go back into your lecture from Monday and correct notes that you might have made there. I'll thank Mark for pointing that out to me. I went to the video to confirm that I had in fact said that, and yes, it was true. So make that correction, please. So, we left off, we were talking about the adaptive immune system. We had talked about the B cells and their role, the T cells and their role, dendritic cells and their role. I thought we would take a step back and look at these cells again in a little bit more detail maybe, or just rehashing the same information in a different way. If we start with our B cells, okay, B lymphocytes, where we find them, we're going to find these cells in the lymphatic system. Generally, they're going to be packed into the lymph nodes, various places within the lymphatic system. They derive from the hemopoietic stem cell that's in the bone marrow. All blood cells come from that, and you can write that down if you want to, but the word that is, but I won't test you on it. All blood cells come from one main stem cell, and B cells go on and mature in the bone marrow. We'll talk a little bit about what that means to mature in the bone marrow, but it has to do with, in fact, determining the antibody that you'll be able to detect. Now, when a B cell is exposed to the antigens, we're talking about them basically setting up residency somewhere in the lymphatic system and letting those antigens kind of wash over them. And so if they have the right antibody, the right receptor, they'll receive or bind to the antigen as they're washing over. But we realize this here. We're talking about 100 million different B cells, and only one of those B cells really is going to recognize any particular antigen that is moving through the lymphatic system. High specificity there. So the B cell then is going to recognize this antigen because the B cell has a surface receptor that is going to be specific for that antigen, right? And we talked about clonal expansion a little bit. So there's 100 million of these B cells there getting exposed to the antigens. One of them will bind to that antigen. So that is clonal selection, the one that gets bound to. And then if the T cell recognizes the antigen being presented and this cell gets activated, then the B cell will undergo clonal expansion. Okay? But remember, clonal expansion here only happens if the T cell okays it. So this clonal expansion that takes place here only going to happen if the T cell is okayed it. So the B cell has internalized this antigen, finds it, internalizes it, and has to present it to the T cell in its MHC molecules. And we'll talk more about those later. So now the B cell, if it is presenting this antigen to the T cell, and the T cell has already seen this antigen before because the dendritic cell presented it to it, then the T cell can activate this B cell and we'll get clonal expansion. And clonal expansion involves two things. Well, this particular B cell just starts to make more copies of itself. And so we get more cells with the surface receptor that's going to recognize that antigen. We get many, many more cells. As this clonal expansion is taking place, the surface receptors that recognize the antigen will be modified here and there a little bit. Modified in that their ability to bind to the antigen can change, such that maybe they start to bind tighter, or maybe they bind looser. There's a looser fit or a tighter fit. And those that fit best will be selected for to continue to expand. And those that don't fit so well, they will not continue in the expansion. So there's a fine-tuning, if you will, as the B cells expand, as this clonal expansion takes place. So now you've got this population of B cells that has a very specific surface receptor for this particular antigen. Two things that are going to come out of this B cell is plasma cells, and we'll start to get many, many plasma cells coming from the expansion of the B cells now. So a lot of these B cells, they turn into plasma cells. They become plasma cells. And what that means is then that they start to release antibodies. So these plasma cells are going to release antibodies that are similar to the surface receptor that first bound to that antigen. And now these antibodies that are being released, and they're all in Y shape, we've talked about that here, they're also going to go out there and they're going to bind to whatever might be having that antigen. So this antibody binds to that antigen there, and now this cell has been labeled. So now a macrophage can come around, recognize this antibody, and engulf this cell. In addition to the plasma cell, some of these cells will become memory cells. And what that means then is when we get exposed to that antigen a second time, these cells will remember that antigen, and having already been activated from a previous exposure, they don't need to present to a T cell again. They can just start to replicate, start clonal expansion without having the helper T cell regulate that. And so Laura asked me a pretty good question at the end of class last time. What about bacterial cells and this notion that sometimes you can get a bacterial infection more than once? Well, this system works the same way with bacteria as it does with viruses. We have vaccines against diphtheria. We have vaccines against pertussis. So we have vaccines that work against bacteria because those vaccines are recognize a very specific antigen on that microbe. Now, with regards to things, bacterial infections that you might get more than once, a couple of things can be going on there. Maybe you were never actually exposed to the microbe the first time or at all. A lot of our infections, are within the digestive tract, and so the microbe's products actually never make it to the immune cells, and so the immune cells don't get exposed. We don't produce antibodies for those cells. A lot of times it's the toxins that are being released, like Vibrio cholera, that causes our cells to release their water and electrolytes and stuff, and that can then cause the conditions that come with Vibrio cholera. So you can get that infection, if you will, or that illness more than once. In addition to that, the microbes themselves can change so that maybe the antibody that we were recognizing changes so that it's a different antibody or antigen, the antigen that we were recognizing changes so that we don't recognize it anymore. And so then an infection could happen. Or if your immune system becomes suppressed. And a lot of times this happens when we get older, our immune system doesn't work so well, and then on top of that we get an illness. And so the illness has then taken attention from our immune system, and so something that maybe at one time we might have had an immunity to, now can take advantage of the fact that our immune system is depressed, and even though we could and should have a response to it, we just don't have the ability to respond to it. So these are some of the ways that microbes can get around our immune system even after having been exposed.
So, clarification, so the plasma cells will continue replication for basically once they've got it forever? So you'll be getting a cell in the body that's, no?
Not forever, no.
Okay.
No. The plasma cells, actually it's the B cells that are replicating, and then they differentiate into plasma cells. Okay, so the plasma cells don't really divide.
So how long is it, I guess what I'm asking is this, the plasma cell and the memory cells, what's the lifespan of these, is what I'm asking.
Okay, plasma cell is going to probably be short-lived, maybe a couple of weeks, depending on how long the infection is. Okay, its main job is to produce antibodies. Okay, as the infection starts to decline, as your body starts to wane, then the T helper cells who are regulating this whole system will start to shut it down. And so the plasma cells will stop producing antibodies, clonal expansion will cease. The memory B cells, you're going to have for the rest of your life, hopefully. And there's another way that it can happen. You lose your memory B cells for a particular antigen. But go back.
So the memory cells, they do replicate then?
Yes.
Okay.
Yeah.
So
that's the only way, because you're not going to have one cell that's probably
they
going to
don't
be able to
live
last
that long.
Yeah. I'm not sure of the lifespan of any specific memory cell per se, but I'm sure it's extended.
Yes.
The memory cells, you said that they're already activated, so they just start replicating early.
Once they get exposed to the antigen, yeah, they don't need the T cells in.
So are they producing then more B cells, or are they producing more memory cells? You only need a certain amount of memory cells?
You mean when they get activated again? Mm-hmm. Okay, when they get activated again, yeah, you pretty much start the whole thing over. Okay, you go through the whole clonal expansion, and you probably get a new population of B cells, because maybe the antigen's changed a little bit, maybe your surface receptor is tightened up a little bit, so you probably will get a new population of memory cells, but not that different. So now let's look at the T cells. And we have two populations of T cells. We have the helper T cells and the cytotoxic T cells. Now, as we've discussed already, the helper T cells really are the ones that are regulating our whole immune response. They first have to be presented with an antigen from a dendritic cell in order to become an effector T cell, if you will. One that can then read an antigen that a B cell is being presented or is presenting to it. So T cell, helper T cell, naive in the lymphatic system for the most part until a dendritic cell comes up and says, here, look at this antigen. T cells, again, are born, if you will, in the bone marrow coming from the hematopoietic stem cell, just like B cells do. But they don't mature in the bone marrow like B cells do. Instead, they mature in the thymus gland. So these guys are going to migrate to the thymus gland, where they're going to mature further. From there, then they'll move into the lymphatic system, and if they've been presented with an antigen from a dendritic cell, okay, and this antigen, just like with the B cell, has to match a specific receptor, surface receptor on the cell membrane of the microbe, or excuse me, of the T cell. So it's going to have to fit in here. The antigen that's being presented by the B cell has to match it. So this means this, right along with 100 million different B cells, we also have 100 million different T cells. So we've got a lot of different cells, and each of them with a specific surface receptor, only going to respond to a specific molecule. So now we've got to get this B cell in contact with that T cell. And that's going to take place again in the lymphatic system. T cell has become an effector T cell because the dendritic cell is presented to it. A couple of things to keep in mind. It's not that the dendritic cell is just presented an antigen that the T cell recognized, but the dendritic cell presented an antigen that comes from a bacterial cell. And how do we know it comes from a bacterial cell? Because the dendritic cell is releasing a co-stimulatory molecule. It's releasing a molecule that tells the T cell that this antigen I'm presenting to you is in fact from bacteria. Without the co-stimulatory molecule, the T cell is going to ignore the antigen that's being presented. And that co-stimulatory molecule is being stimulated because the dendritic cell has toll-like receptors, just like the macrophage does. And those toll-like receptors, which bind to peptoglycan, flagellin, and those things, have been bound. So the dendritic cell has its toll-like receptors bound by bacterial antigens, and it is presenting an antigen that probably has come from that same bacterium. And so this then alerts the helper T cell that this is something that we need to be aware of. And now that helper T cell is an effector helper T cell.